It is really important to score the alignments in order to have an inference about the strengths of those similarities. So there are different scoring schemes which have been introduced. So scoring in general is done in this way, like we take the score for each aligned pairs, whether they are matched or mismatched, and we take that score, and in the end we add the gap penalties. For every gaps we do some penalties, we call it as gap penalties. So here we have in the model we have sum of score of x comma y, xi, yj. We are having 2 pointers. I is running on one strand and j is running on the other strand. We will talk about when we look into their algorithms. And then we do some add some terms for the gaps. So basically the gaps are actually, the scores are in minus, so actually these are penalties. So that score is, in real sense, that score is subtracted. Here if we have a gap penalty, so that stays as constant, we call it as linear gap penalty. We have another type of gap penalty which we call it as affine. We'll talk about that in a later section, but for now onwards you should remember that for every gap we will penalize with the same way, or with the same penalty. Like if it is -2, it says same throughout. So this kind of penalty is called as linear gap penalty. A simple scoring scheme may be to score an alignment is to count 1 for each match and 0 for mismatches. For instance, if we look here, we are aligning two sequences, and those vertical lines are indicating where we get the alignments, and gaps or blanks means mismatches. So in this case, we calculate those vertical bars to be 12. So the score of this alignment is 12 versus another pair down there where we see those 5 matches. So the score is 5. Scoring or substitution matrices are really important concepts, are really important matrices which are used for scoring amino acid substitutions. Basically, in the proteins, we observed that some amino acids they replace the others most often, and some of them they are less frequent. So the scientists, they came up with some scoring schemes, and they presented in the shape of those matrices. We call them as substitution matrices. So they reflect substitution rates that are originated by evolutionary events. So they kind of try to look into their evolutionary relationship and how often or how less frequent those substitutions are there. So in this way these matrices are built. Some of the matrices to compute those sequence alignments, one of them is PAM, point accepted mutation. The other one is BLOSSOM, block substitution matrices. We'll talk about them in a later section. For a set of well-known proteins, what they do, what these substitution matrices they believe, so how they are formed. So we align the sequences and we count the mutations at each positions, and for each substitution set then they calculate the log-odd ratio. So we get some data sets, and from that data we calculate how many times amino acid A has been substituted by B, and then we compare it with their expected ratios, and then we convert it into the log scale. So in this way we come up with these scores. We call it as log odds. So it is log of observed or expected. So if we have the upper term higher, it is more frequent, obviously our score will be positive, and if lower term is higher, we see them less frequent than their expectations, then the scores they become negative. So a positive score in those substitution matrices indicate that amino acids are more similar, and it is the presence or the substitution of one amino acid to another is more frequent than compared to the expectations or expected by chance. So in this way we will get a positive score. Whereas if the amino acids are dissimilar and mutations from one to another they occur less frequently, so in that case the score is negative. So this is how they came up with log-odd PAM 250 matrices. We don't have time to go into the details how they are constructed, but they get some data sets, and from that they look into their observed those values or observes substitutions and compare with their expected ones, and then convert them into this log scales. Basically it's 20 by 20 matrices, but we are just looking into half of it, so it is reduced into half because we don't want this information to be duplicated. So we just take part of it. So if we look into its top, so how many times this serine is seen, so the score is 2. How many times this serine is, if we look into this row, we see this serine right here, and how many times it's been replaced by threonine, so that is, the score is 1. So in this way we can align two pairs, and even if they are not the exact identical pairs, we can score them by using these similarity matrices. So for example, we have a raw score of an alignment. We get T, which is a mismatch with A. We get that matrix and we get the score, which is 1. P is exactly matched with itself. The score in the matrix was 6. So in this way we can add their individual scores of these pairs and put them together while deriving these scores from the matrices. We are keeping in mind their evolutionary history. So in this way this alignment scores 9. So these substitution matrices are log odd matrices which are used for scoring amino acid substitution in pairwise alignment. So these use of these substitution matrices is a kind of a better approach where we can keep in mind the evolutionary relationships between two amino acids.